Science is fun. It's a way to be creative. Oh, under. Okay.
Okay, now he's in this spot.
Oh yeah, he's cornered. Oh.
Oh! I'm not getting my net down fast enough.
Okay. I can think of any idea or any prediction that I think the world acts, and then I can go out and test it. It's even more fun when my predictions aren't true, because then you learn something that you would have never thought about. It's always changing. There's always new things that you're learning, and there's so much about the natural world that we don't understand. Hi, my name is Suzanne. I'm a graduate student at UC Berkeley, and I'm studying life histories of Pacific salmon. We're looking for steelhead. It's Oncorhynchus micus. It's the only fish in this stream, so fortunately when we see a fish, we know it's our fish. In California, we find primarily two types of Omicus. One is known as the steelhead trout, which is the migratory form. So it's what we think of as a salmon, where it spends half of its life in the ocean feeding and rearing. And the other form is the resident form, or rainbow trout, and it spends its entire life history in freshwater. And both of these life histories, although they're very different, they co-occur in coastal streams in California, and so I'm interested in what maintains both of those life histories and how they both make a living following these different strategies. Yeah, they're hard. Can you see this guy in the water right here? He's right here. He's really camouflaged. Do you see it? So these little ones that we're picking up right now will be in the freshwater for like 2 or 3 years. And then they'll go out to the ocean for another 2 or 3. And so then the adults, when they're ready to spawn, they'll come back and they'll swim upstream. So that's the migratory form, and then the resident form mature in freshwater. So they never leave. It comes down to, should I stay or should I go, and who's going and who's staying, and how is that related to their feeding and growth behavior. And so I'm using a variety of different methods to understand how these life histories survive in the stream and what causes both of them to coexist. So over the summer I tagged a bunch of fish, similar to the technology that you use to tag a pet, where you implant it with a little chip, or also the same technology that you use to check library books in and out.
That's how we estimate the population, is by marking them with these pit tags and then figuring out what proportion of the population you recapture. And by feeding that into a computer statistics program, you can get an estimate of the population size. And we just insert this really small passive integrated transponder that we can read with these scanners. Basically like a barcode, like scanning your grocery item.
So I've chipped a bunch of fish, and I can recapture them and detect them as they swim out to the ocean to understand which individuals are making that migration to the ocean, and then recapture individuals and understand which ones decide not to leave. And then the second aspect is looking at their genetics. So the life history is a heritable trait. If your mom was a rainbow trout, you're more likely to be a rainbow trout. And so I'm genotyping a bunch of fish to understand variation at a certain chromosome that we know is linked to life history expression. If we sample only brothers and sisters, then it biases our results towards that specific family and what their genotype are. So that's why we're trying to sample, cover more ground, even though some pools are really easy for us to catch fish and other ones are harder. We try to move on and get fish from every pool, so we're not all sampling from one family. And so we're taking these fin clip samples to use a genetic marker that has information relative to that life history. So we can assign a genotype to each individual that we take a fin clip from, and then make some inferences about its life history and its parents and its ecology from that. So I go to a genetic specific lab to fragment it. So you chop it up into little pieces and then you sequence those little pieces. But part of the decision on whether they should stay or go is the age of fish. So when we're in the field, in addition to taking genetic samples and tagging the fish, we also take scale samples, and we can use the scale samples to age the fish. So similar to how trees lay down rings as they grow, the fish lay down these rings on their scales as they continue to grow. The gap between these spacings are smaller in the winter, and then they're wider in the summer when it's warm and there's lots of food. It's important to understand how old they are to know if they've actively made that decision to stay as a resident, or if they're just still feeding and rearing in the freshwater and they haven't out-migrated yet to the ocean. The first thing was to know for the creek that I'm working on if both of these life histories do occur, and we predicted that we would find both of them in the stream. The second result that we've gotten back so far is where they physically occur in the stream. A lot of these coastal streams have lots of waterfalls, which are barriers to adult fish who are migrating upstream to spawn and rear in these freshwater streams. And so one of the things that we found was that the resident fish were primarily found very far upstream in these creeks and above these waterfalls that are somewhat impassable to the migratory adults. And then downstream near the mouth of the creek, we found that the region was dominated by mostly migratory genotypes. And then we're still getting some mark and recapture data back, so still detecting fish and recapturing fish to understand if those genotypes do actually predict the life history phenotype that we see. The waterfalls are a barrier to the upstream migratory adults, but they're only a barrier in dry years. So if there's many rainstorms and many flow events in the stream, then the migratory adults generally have a much easier time ascending that waterfall. We're predicting that in these really wet years, the migratory form and the resident form have many more opportunities to interbreed, and that they're found in the same pools and in the same reaches when the migratory adults have the opportunity to get above these waterfalls. The backside of that is in the dry years, there's very few opportunities for these migratory adults to make it above these natural barriers, so we find that there's more of a spatial separation. And looking forward, we are expecting to see more drought in California, and that does have implications for the system where we find this spatial separation of these two life histories is dictated by waterfalls and precipitation. So if we see less precipitation, we'll see increased separation of these two life histories, and maybe in the long run we would see speciation, but for now it would just mean a lack of gene flow between the two and a lack of interaction.
Slow your breath down, just take it slow. Find your smile now. Whoa. You can try.